Okay. So lecture 12 is very important because it is about the different types of tissues that we have. And histology is the study of the tissues. Remember, we learned in the past that anatomy was only at the level of the cadaver, the anatomy of what we are able to see, the anatomy of the. It's funny how you guys are looking at the camera. You're not looking at the slides. Okay. I'm talking about the tissues and everyone is looking at that little window up there. I'm not gonna have it anymore. Especially Shayma. She's looking up there. Okay. That's hilarious. Okay. That's morning hysteria. Morning laughter hysteria. Okay. So the tissues are what make up the cell, the what makes up the systems, the organs, and we said that we don't have organs. We don't have the heart system. We have the cardiovascular system. We don't have the intestine system. We have the digestive system. We don't have the lung system. We have the respiratory system. But these systems are made up of organs that are connected, interconnected together, physiologically, anatomically, structurally, functionally. And we also got to learn today that back in the day, there was gross anatomy, where we examine the cadavers, what is seen with the naked eye. And that starts from the organ and above, organ system, and the organism and the population. But when we study what is not seen with the naked eye using a microscope, that is histology. When we investigate the tissue histology and cellular histology. So tissues are a group of structurally and functionally related cells and their external environment that perform a common function. So we cannot isolate the cell from its surrounding environment because the same way we are a product of our environment, so is the cell. So the cell who is in exposure with the atmosphere, with the ultraviolet radiation, the cell that is in exposure with these microbes that are in the air have to have a structural support that enables them to adapt to all of these threats that surround the cell. If we have a different type of tissue that is more in the viscera, that's inside the body, that does not deal with outside external environment threats. It deals with internal environment stress, a different form of stress. So they need their own structure and function. Okay, so the tissues share two basic components. The tissues are a group of structurally, anatomically, and functionally related cells, and they have an extracellular matrix. This extracellular matrix is the society. Remember, we talked in the past that the cell is a community, is a society that is compartmentalized. There are different regions. Gertrude, I'm not going to accept it if you don't hand it in now. You're going to get a zero. I'm not going to accept it. Right now, you have to submit it. I've already waited a lot. No, I cannot wait any more than that. We are done. Can you please hand it in? Okay, zero. All right. So we move on. There are different types of tissues. So we have four types of tissue. There's epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So these are the four tissues you need to have in mind. So you're supposed to be able to reproduce. What are the four types of tissues? Epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So you must know these four guys. You must know the epithelial tissue, the connective tissue, the muscle tissue, and the nervous tissue. So there is no other type of tissue besides these four. Okay, so two of these tissues, the epithelial tissue and the connective tissue, these two types of tissues are not excitable. They do not respond to membrane voltage change. They do not respond to membrane voltage change. While the muscle and the nervous tissue, these types of cells are excitable. They respond to membrane voltage change. So that is the first distinction that you have to have in mind. These last two, the muscle and the nervous tissue, both of those are non-excitable. Both of those are not responding to membrane voltage change. How can a cell be excitable? How can a cell respond to a membrane voltage change? It is by the opening of these ion channels. Remember in the past when we talked about ion channels opening, and the ion channels, when they open, they allow for the influx. Who remembers what influx is? Influx. Yes. Excellent. So influx, efflux, exit. So when the electrons exit, so when the electrolytes, the potassium exit the cell, that's efflux. When the sodium enters the cell, that's a form of influx. So this influx and this efflux is causing a membrane voltage change. That membrane voltage change excites the cell. So when the muscle cell and the nervous cell get excited, both of them are excitable. Are these two excitable? Are the epithelial and connective tissue cells excitable? No. No, they're not excitable. So that means membrane voltage changes will not be detected by connective and epithelial tissue. But even though both muscle and nervous tissue are excitable, does that mean once they get excited, they do the same thing? If you get paid $1,000 and I get paid $1,000, are we gonna spend it the same way? No. So the same thing with these excitable tissue. The excitable tissue is as far as the muscle cell. When the muscle cells gets excited, what happens? It contracts. When the muscle cell gets excited, it contracts. But when the nervous cell gets excited, it does not contract. The muscle cell, when it gets excited, it does not contract. What happens? It relays a signal. It mediates the transmission of a particular signal. It radiates, it mediates an input. Remember when we discussed homeostasis, we discussed an input, efferent neuron carrying an output, afferent neuron carrying an input, integration. Remember the computer integrating the information. What does that lead to? That leads to mediation of the information. So when the skeletal muscle cell gets excited, it does not mediate input or integration or output. What does this muscle cell do? What does the muscle cell do? Contract. What does the nervous cell do when it gets stimulated? Okay, excellent. Okay, so epithelial tissues. Epithelial tissues are, this is the first type of tissue. Remember, we got four types of tissues. What are the four types of tissues? Who can remember for me what are the four types of tissues? Two are siblings and two are not. The first one is connective. Excellent, excellent. So let's begin with the first one, the epithelial tissue. So epithelial tissue is sheets of cells with no visible extracellular matrix. So now you're talking about their features. You're describing their histological structure. You're describing your histological structure. So these are sheets of cells with no visible extracellular matrix. And they cover all the body surfaces and the cavities. So remember, we are hollow from outside and we are also hollow from inside. We're hollow from outside, we're hollow from inside. And what happens is the sheets of the cells are intact, okay, and they are providing a protective layer. So for example, your outside is very similar to your inside. This elementary tract, this hole is very similar on the outside to the inside. and we are hollow from outside and we are hollow from inside. There's a cavity surrounding us and a cavity in the inside. So when you take some food and some food does not get absorbed, like people who have lactose intolerance, that milk they drink, they cannot absorb it into their blood. So they have diarrhea. So that stuff they excreted actually technically never made it in their body. Because just because it got into the stomach and into the intestine, it didn't make it into the blood. So it's still outside the body. You are outside until you make it into the blood. And these tablets that people take, the bioavailable fraction of that tablet is like 5%, is like only 5 to 10% makes it to your blood, and 90% gets excreted. So that 90%, although it made it to your body, but it was never absorbed. It was never absorbed. Okay, okay. So there are specialized cells that form glands. So this epithelial tissue is the type of tissue that forms the gland. So you know if you have the endocrine gland, the hypothalamus, if you have the ovaries, the testes, these are all glands made up of what kind of tissue? Connective tissue, muscular tissue, nervous tissue, or epithelial tissue. So that's the type of tissue that is making your gland. That's the type of tissue that is lining your skin and your cavities from inside. Okay, the connective tissue is different from the epithelial tissue. The connective tissue, from its name, it connects other tissues to one another. It connects other tissues to one another. And it must have a large extracellular matrix with cells scattered throughout it. With cells scattered throughout it. So this is the ocean that surrounds the island. The island is your tissue, your cells. But the extracellular matrix is the ocean that surrounds them from the outside. And they have these connective tissues have large extracellular matrix. They bind and support and protect and they transport substances among the tissues. The muscle tissues are different from the connective tissue and the epithelial tissue. They contract and they generate force. They contract and they generate force, and they have a very little extracellular matrix, and they include smooth cardiac and skeletal muscle. So remember, people think that muscle means that the skeletal muscle means your skeletal muscle. No, you have your smooth muscle, your blood vessels. What's an example of smooth muscle? That's your blood vessels. No, 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 no. Skin, that's the epithelial tissue. We're talking about muscle. We're talking about muscle tissue. So muscle tissue is three types. There is smooth muscle, there is cardiac muscle, and there's skeletal muscle. Okay, the nervous tissue. The nervous tissue has a unique extracellular matrix. So the cells generate, send, and receive messages. We said that the cells receive input, they send output, and they generate, meaning they integrate this information. They include other cells that support the function of the nervous tissue. Okay. Extracellular matrix. Extracellular matrix is for substances, it's describing to us substances that surround the cells. So what is the structure and function of the extracellular matrix? The extracellular matrix provides strength to resist the tensile and compressive forces. So we need a scaffold. We need a steel girdle that protects the building. So we're going to need this extracellular matrix that protects the tissue from shock, that maintains for the tissue osmotic balance, electrolyte balance. And the extracellular matrix is also important, not just to give tensile integrity for the tissue, but also to direct the cells to proper place in the tissue, and it regulates the development the mitotic activity, and the survival of the cells. And it holds the cell in proper positions. It anchors the cell in place and participates in this cell cycle or cell division cycle. What are the components of the extracellular membrane? The extracellular membrane has two components, the ground substance and the protein fibers. Now let's talk about the ground substance. The ground substance contains extracellular interstitial fluid consisting of water, solutes, and macromolecules. So outside of the cell, you have your extracellular matrix. This extracellular matrix is very important, but in order to understand the significance of the extracellular matrix, we have to divide it structurally into two components, and that is the ground substance and then the protein component. The ground substance is everything that is not protein. Everything that is not protein, which includes the carbohydrates, the lipids, the water, and the solutes. This is the ocean that's around us. So we have in the ocean, there is fish, there is water, there is salt, there's electrolyte, there's different substance, and the cell also is surrounded like an island with water and solutes and macromolecules. So here we go. So where is our extracellular matrix? Do you think it is this really big ocean, or do you think it's this one? So what is this here that I'm pointing to with my mouse? That's the cytosol. So this is obviously my cell membrane, and these are my integral proteins. So these integral proteins, we refer to them also with another name. What's the other name? What's the other name? Transdimensional. Transdimensional. This is a nice name, but you almost there. It's not transdimensional. What is it? Transmembrane. So these transmembrane proteins, they remember they call them transmembrane because they traverse both layers of the membrane. And what happens on the extracellular membrane? In the extracellular membrane, you see you have two different components. You have your ground substance and you also have your protein fibers. Who can tell me what do we see now in this image? Who can identify for me where is my extracellular matrix? Everything that is not the cell, everything that is not here, that's not the cell. But everything that's outside, that's part of the extracellular matrix, because you see the extracellular matrix is two things, ground substance and protein fibers. So you have the ground substance in the extracellular matrix where you have glycosaminoglycan. These are glycosaminoglycans, and these are proteoglycans, and these are proteoglycan aggregates. You see this proteoglycan. Proteo means protein, glycan means sugar. But that is not a glycoprotein. Glycoprotein is attached to the cell. So if I ask you what's the difference between a glycoprotein and a proteoglycan, a glycoprotein is a sugar that attaches to the proteins at the surface of the cell membrane, like here, like the green who attaches to the protein, like this one, that's glycoprotein. You see, that's glycoprotein. But proteoglycan is more attached to the extracellular matrix. And these are proteoglycan aggregates, and these are glycosaminoglycan. So, what are glycosaminoglycans? From the name, glyco means sugar, amino are the building blocks of proteins. So, these are compartments that put for us the structure of the extracellular matrix. And the extracellular matrix is, you have to appreciate that it is solute, it's fluid, it is fluid ocean, it has electrolytes, okay, it has the ground substance, which is everything that is not protein fiber. So these are your proteoglycans, these are your glycosaminoglycans, and these are your but your collagen fiber, now that is not ground substance. That is protein fiber. your reticular fibers, and these are your elastic fibers. Okay, ground substance. Ground substance is three families of macromolecules known as glycosaminoglycans, proteoglycans, and glycoproteins. These are the three families of all the macromolecules that make up your extracellular membrane. Remember them. Glycosaminoglycans, known as the GAGs, glycosaminoglycans, and proteoglycans. These are proteins with sugars, proteoglycans, and glycoproteins. Glycoproteins. And then when we learn about the protein fibers, we're going to see that it is made up of collagen and made up of these reticular fibers and elastic fibers. These are all proteins. The last one is glycoprotein. Glycoproteins. Okay, so glycoproteins are on the surface of your cell, and these, you see, glycoproteins, proteoglycans, and glycosaminoglycans. Okay, glycosaminoglycans. Glycosaminoglycans are polysaccharide chains with negative charges that attract cations to draw water out of the cell, to draw water outside of the cell. So that's the first significance of the glycosaminoglycans. These are polysaccharide chains with negative charges that would attract the cations to draw the water out of the cells and the blood vessels. So in osmotic balance, we draw water from inside of the cell to the extracellular matrix through you and I paying attention through glycosaminoglycans. These glycosaminoglycans here, they are able to draw. Remember, we said in diffusion that these solutes, are you guys following? These solutes are able to, through the process of osmosis. So that's how water gets out from the cell into the tissue. That's how water goes from the cell outside into the tissue. Okay. So the polysaccharide chains have negative charges that would attract the cations to draw water outside of the cells and the blood vessels. So what is the name of the process that mediate that? Thank you. Very good. Okay. It's a long name, but now you know what is it part of? Is it part of the ground substance or is it part of the protein fibers? The ground substance. Okay. So proteoglycans. Proteoglycans form aggregates such as hyaluronic acid, which makes the tissues more solid and resistant to compression. So now the job of proteoglycans is different from the glycosaminoglycan. That one was important for water osmosis. That is important for regulating the osmotic balance. But proteoglycans form aggregates such as hyaluronic acid, and it makes the tissues more solid and resistant to compression. Glycoproteins. Glycoproteins are also called cell adhesion molecules that glue cells to each other. Cell adhesion molecules that glue the cells to each other. You know that these are your CAMs and CAPs, cell adhesion molecules and cell adhesion proteins. So they hold the cells really tight together and they bind the cell surface proteins and the protein fibers and proteoglycans. It maintains the architecture of the tissue. Now we finished with the ground substance. Let's look at the protein fibers. So now this is no longer the ground substance. This is protein fibers. This was the ground substance. You see, the extracellular matrix is composed of ground substance. So the extracellular matrix, the extracellular matrix Okay, guys. Extra cellular matrix, extra cellular matrix is divided into two parts. The first one is known as ground substance, and the second part is known as protein fibers. Okay, ground substance is made up of three parts. We said the first ones are the gags, known as the gags. Oh, hold on, I got it. Glyco, amino, glycans. Excellent. The second part, what is the second part? These are your proteoglycans. And the last part is glycoproteins. Glycoproteins. Now you got to know what they do. These ones are important for water balance, osmosis, and the proteoglycans are important for form aggregates such as hyaluronic acid. But what does that do? Makes the tissue solid. Yeah, that gives tensile integrity. So if we go back here, that's what they do. You see? First, we learned about GAGs, the glycosaminoglycans, and they are important to draw water outside of the blood vessels, and they form aggregates such as hyaluronic acid, and they make the tissue more solid and resistant to compression. So that's what proteoglycans do here. And lastly, you have your glycoproteins, and these are cell adhesion molecules that you see CAMs, cell adhesion molecules or cell adhesion proteins, CAPS, that glue the cells to each other. It binds the cell surface proteins, the protein fibers, and proteoglycans. It maintains architecture of the tissue. They maintain architecture of the tissue. Yeah, glycolipids. So you're talking about glycolipids. So glycolipids are part of your cellular immunosurveillance agents. These are the ones we use to detect this self from non-self. But the extracellular matrix is composed only of these three. So glycolipids is not part of them. Now the other part is your protein fibers. We are here now. So the second part of the extracellular matrix includes the collagen, elastic, and reticular fibers. It's made up of collagen, elastic, and reticular fibers. They're all fibers. That's why we call it protein fiber. Collagen fiber, one. Elastic fiber, two. Reticular fiber, three. Okay, let's begin with collagen fibers. Collagen fibers constitute 20 to 25% of protein in the body. And they appear like an entwined steel cable, and they are therefore resisting tension and pressure. You see the collagen fibers? They are like wires and steel cables that are intertwined, and they hold they are resistant to tension and pressure. So they keep the tissue from being deformed and affected by the pressure. Elastic fibers. So elastic fibers stretch without breaking, with extensibility, and they return to the original length or the elasticity, and they are formed with protein elastin. They are formed with protein elastin. So elastic fibers are formed by which type of protein? Elastin, from the name, elastic, elastin, and stretches without breaking, and they return to the original length. They return to the original length. Reticular fibers. Reticular fibers are a type of collagen that is thinner. You see the reticular fibers. These are your reticular fibers. They form a meshwork to support the cells and the ground substance. And they have webs. Webs. These webs trap the foreign cells. So you can tell that these reticular fibers have immune defense activity because they are the ones who trap the foreign cells. Do the elastic fibers trap the foreign cells? Do the collagen fibers trap the foreign cells? It is only the reticular fibers. Okay. So here we see that the collagen, this is the big purple one, that's the collagen. And this here is reticular fibers. These are your elastic fibers, these thin pink ones, and these blue ones are your reticular fibers. And they are the ones who are like a net. They trap these immunoparticles. We talked about junctions before, and they are between the cell. So they anchor, they are like staple. They anchor neighboring cells through integral proteins. So you see, these are our cells. There are three types of junctions. It's very important to know that. The tight junctions, the desmosomes, and the gap junctions. So the easiest one to know are the gap junctions because they are tunnels, they are gap. That's why they call them gap junctions because through them the cells pass to each other's stuff. Okay, back in my hometown, we have a back door that is connected with all, so the homes, they are not like here. There's like a garden and a front yard and a backyard and then a fence and another home. No, the homes are like this classroom. They are like blocks. So there are sometimes door between neighbors to neighbor, and people like share stuff. People like send to each other stuff. So there's the outside door. You can get out and move through that door, and then there's like a back door that people can exchange stuff. This is what's happening here in the cell. You see, the cells have these back doors. These are known as they are gaps. They allow things to flow. Solutes will pass through. Electrolytes will pass through, and molecules will pass through. Are we clear? Okay. Then there are desmosomes. Desmosomes. You see these desmosomes, they hook. They hook the cells so tightly. They hold the cells from separating. And they hold them so tightly that they do not even allow water to leak. And there are also gap junctions, tight junctions. That's why they call them tight junctions. They're clipping the cells so tightly even water cannot seep through. So now let's examine each one of them. But you got to know your junctions. We have three types of junctions. We have three types of junctions. Oh, amazing. We have three types of junctions. Oh, no. So three types of junctions, of cell junction. There's the gap junctions. There's the tight junctions, gap junction, tight junctions, and there are the desmosomes. So you have to label those three. Now let's investigate them. So the tight junctions, they hold the cells tightly like a zipper. That's why they call them tight junctions. And some are leakier than others. And desmosomes, they are external fluid can pass between them. These are more loose. That's why we don't call them tight junction. And they increase the strength of the tissue. They increase the strength of the tissue, and they have integral or linker proteins that attach intermediate filaments of the cytoskeleton. So which of the three cell junctions is actually associated with your cytoskeleton? Is it the gap junctions? Gap junctions are not attached to your cytoskeleton. Is it your tight junctions? Look here. Are the tight junctions attached to the cytoskeleton? No. Then who's the only one who's attached to the cytoskeleton? These are the desmosomes. that are attached with integral linker proteins that attach to the intermediate filaments of the cytoskeleton. And these are your gap junctions. You see they are tone. These are the easiest one to understand. They are made up by different proteins that they get stuck and then they make a hole. So the proteins look like that. That's protein number one. That's protein number two. That's protein number three. That's protein number four. One, two, three, four. Then they would assemble together to make for me this whole. One, two, three, four. And they all assemble together to generate for me this whole. So these are the, you see the different proteins. One, you see one of my mouse is one, two, three, four, five, six, and they all assemble together to allow the passage. You see from one cell to another. They allow the substances to pass from one cell to another. There are tight, there are also, now let's talk about epithelial tissue. The epithelial tissue is found on all the body surfaces. So based on this figure, what can you say about epithelial cells? So these are your epithelial cells. This is your connective tissue, and you see that the blood is actually on your connective tissue. The blood is found on the connective tissue, and you have the basement membrane. The basement membrane is made up of the basal lamina, and you see the basal lamina is closer to your epithelial tissue, and then there's the reticular lamina that is closer to the connective tissue. And the middle layer is the basement membrane. Then the upper layer is your epithelium. And this epithelium has an apical side. This apical side is free side, and this is the basolateral because it's attached to the basal lamina. Please remember these three layers. Please remember these three layers. The top layer is your skin epithelial tissue, and that's the air. The air is out here. That's the air, and that's your epithelial tissue. And this is your basement membrane divided into two layers, the basal lamina closer to your epithelial tissue, and then below the basal lamina, you're still in the basement membrane, but this is not the basal lamina, that's your reticular lamina, and below it is your connective tissue, where there is blood vessel. So if I ask you on the exam, where do you see blood vessels? Do you see them on your epithelium? Do you see them in the basement membrane? Do you see them in the connective tissue? Excellent. If I ask you on the exam, what are the two layers that make up the basement membrane? The basal lamina and the reticular lamina. Okay. And what is the side that is facing your basal, that is facing your epithelial cells? This is your apical side. Apical side. This is your these lateral surfaces are here. These are the lateral surfaces, the boundaries. But I'm talking about here where my mouse is. So that is this is the outside. That's the street. This is your cavity. Okay, these are the cavities. Either the cavities of, so for example, your skin. Your air is the apical side. So that's air. So you have air here, you have your apical side, then you have your epithelial tissue. Below the epithelial tissue, you have the connective tissue that is divided into basal lamina and reticular lamina, and then below that is your connective tissue. This is very important to have a mental picture of this. Okay, what are the functions of the epithelial tissue? The functions of the epithelial tissues are protection. They are there to protect. They are there for immune defense because there are a lot of resident sentinel macrophages, dendrites, immune cells that would attack, immune cells that would detect the presence of any pathogen and destroy it. There's a secretion, transport, and sensation. So let's begin with protection. So from mechanical and thermal injury. So your skin is important to protect you from mechanical and thermal injury. That's why this tensile strength, in the extracellular matrix surrounding your epithelial cell. This very important, this very important, um, uh, this very important layer is there to protect you from the heat. The sun is always flowing, and the ultraviolet radiation is always striking your skin. Ultraviolet radiation is always striking your skin. Now, your skin has to be very strong to withstand this heat. So, this epithelial tissue is very strong. Otherwise, it will not be able to withstand the sun. Same thing from inside, from your stomach. Your epithelial tissue, remember, epithelial tissue is not only outside. I began the lecture telling you we are hollow from outside and inside. That's why the food we take that is not absorbed is actually never made it to the body. So imagine your stomach has a lot of acid in it. Why isn't this acid burning your own stomach? Because that epithelial tissue is very strong and it's a paste. It's from the inside and on the outside as well. Um, Also, your skin has a lot of acid known as acid mantle. This acid mantle is like a lava layer where if your any bacteria or virus or parasite or fungi attack your skin, this acid mantle is going to burn it. But your own skin needs to be protected from that acid. So that's why the epithelial tissue is very strong. Keratin. Remember when we talked about the different types of protein structure? There is primary structure, there is secondary structure, there is tertiary structure, there is quaternary structure. We said that keratin was what structure? Keratin. Keratin was? Keratin was what structure? That notebook is not going to be on the exam. It has to be inside the head. Secondary, excellent. So keratin and who else? Collagen, excellent, Brittany. Excellent, excellent, excellent. Keratin and collagen. That's secondary. Who is tertiary? Three. Hemoglobin is quaternary. It has four subunits. It has two alpha and two beta. That's four. It's quaternary. Who is tertiary? Who's the brother of hemoglobin? Myoglobin. And we said, what's the difference between hemoglobin and myoglobin? Stores it. Excellent, 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 excellent. Very good. Okay, so the skin is subject to damage but has high mitotic rate. So this skin, this epithelial tissue, is able to enter the cell cycle. This epithelial tissue is able to enter the cell cycle. So not every particular type of tissue is able to enter the cell cycle. Your skin can. That's why when you have a scrape or when you have a scratch or a bruise, your skin can heal. That is known as regenerative tissue. It can regenerate. So when we look at the cell cycle is divided into two big phases. What are the two big phases that comprise the cell? The first one is known as mitosis, and there's interphase. 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 Interphase is divided into three steps. You have your G1, your S phase, and your G2. Then after that, the cell will go through G1, doubling in the cytoplasm, doubling the organelles, then goes through the S phase, doubling the DNA, then goes through the G2 phase, preparing to enter mitosis, checking that this DNA is not going to be damaged. So we don't want to replicate a damaged cell. We don't want to replicate a cell with damaged organelles or cytoplasm. Then we enter mitosis, and in mitosis, we're going to have prophase, metaphase, anaphase, telophase and cytokinesis. So to enter this cycle means you're gonna birth, develop more cells, more cells, more cells. So your skin can do that. If I scratch my skin now, it's gonna enter the cell cycle to build and heal. That's known as healing by regeneration. Regeneration. Okay, it's funny. Regeneration. Okay. Starfishes after they lose a piece of cells, they regenerate too. Yeah. So regeneration, that means their cells. Your liver. You don't have to go all the way to starfish. Your own liver. If you cut, when they do liver transection and they cut the liver is gonna build up again and gonna make up for the part that was lost. So that is regeneration. But some other tissue cannot regenerate, cannot enter the cell cycle, like your neuron. If your neurons can regenerate, we will have no Alzheimer's disease. The 90-year-old people will be going to college studying math and computer science. But it's not going to happen because your neurons, that's why when you have a stroke, that part of your brain is gone forever. If it wasn't the case, then you'd have a stroke, then that part would die, and then a new neuron would form. That's not what's going to happen because that neuron tissue is non-regenerative. It cannot enter the cell cycle. So it heals not by regeneration. It heals by scarring, scar tissue formation, fibrosis, healing by fibrosis. The immune cells scattered throughout the epithelial tissue. Okay, secretion. They form glands that produce substances such as oils or hormones. There are substances that are secreted through a duct or into the bloodstream. They are secreted into a duct or into the bloodstream. We already know what type of tissue is responsible for gland secretion. What type of tissue is that? Is that muscle tissue, nervous tissue? Is that connective tissue, or is that epithelial tissue? Epithelial. Okay, so transport into other tissues. The epithelium is selectively permeable. And the substances cross by passive or active transport. And they are critical in areas such as the small intestine, critical in the small intestine. The epithelia are highly innervated. The epithelia are highly innervated and they detect changes in the internal and external environment. They detect changes in the internal and external environment. So what are the features of the epithelia? The epithelial tissue are avascular. That's not secret. What does avascular mean? Avascular. Is it like no blood flow? Good, very good, excellent. So that's not a secret no more, right? Because we've seen that the epithelial tissue has no blood vessels. It's avascular. What about the basement membrane? Does it have blood vessels? No. Who is vascular? Later on the exam, they're going to ask you which one of the four layers is highly vascularized. Is it the epithelial tissue? Is it the bone tissue? Is it the nervous tissue? It is the connective tissue. That doesn't mean the others don't have vascular supply. They do, but the connective tissue has a lot. And this is where you achieve your thermal regulation. So when these blood vessels dilate, when you're so hot, remember we said in hyperthermia, when you're so hot, the blood vessels near your skin would vasodilate to accommodate more blood so the blood can flow out from an area of high heat to area of low heat. Okay? So that shows the blood is going to leave, but the blood vessels are not up here. They are in the connective tissue close to your epithelium. Okay, functions of the epithelial tissue. Okay. All right, so now let's look at the features of the epithelia. Epithelial tissue are avascular. That's not a secret anymore. You see, these are the blood vessels down here in the underlying connective tissue. They are not up here. And the basal lamina. The basal lamina is the basement membrane. The basal lamina is the basement membrane, and this is the epithelium. So that's our epithelium. These are our tight junctions. These are our desmosomes. Who's missing from this diagram? Gap junctions. Very good. Then below the epithelium, you have, what's the name of this upper layer of the basement membrane? Is it the reticular or the basal lamina? It's the basal lamina, then reticular lamina, then your connective tissue that is full of blood vessels. So there is no extracellular matrix between the epithelial cells. You see, they are so tightly closed, no extracellular membrane between them. The extracellular membrane is present below the cells as a basement membrane. Is present below the cells as a basement membrane. So that is the extracellular membrane. It's reversed. It's flipped upside down. So usually in all the tissue, the extracellular membrane is on the outside, but here is actually reversed. It's actually underneath because the skin needs to be out. Needs to be. Imagine you have a basement layer above your skin. Then your body is not going to be able to do its function because now your skin is not in intimate relationship with your extracellular environment. So here we reverse it. But in the rest of the body, usually in the rest of the body, the extracellular membrane, the extracellular membrane the is usually beneath and between the tissue cells. So the basement membrane has an upper basal lamina and a lower reticular lamina. The basal lamina contains extracellular membrane that is synthesized by the epithelial cells. So this is the basal lamina. It has extracellular membrane that is secreted by so these epithelial cells, they keep on squeezing like toothpaste. So all of this basal lamina is squeezed by your own epithelial cells. These cells, they are the ones they squeeze out this basal lamina. And the basal lamina is consisted of collagen and ground substance. And we know that. We know that the extracellular membrane is always ground substance and protein fibers. Now, the reticular lamina, that's beneath your basal lamina and above your connective tissue. So the reticular lamina is made by deeper connective tissue. It's made by deeper connective tissue, and it consists of reticular fibers and ground substance. Consists of reticular fibers and ground substance. You see, these are the reticular fibers, and this is your reticular fibers and your ground substance. So ground substance is found in both. Ground substance is found in the basal lamina and ground substance is found in the reticular lamina. Then what is the only difference? The only difference is that the fibers. So the ground substance is the same, but the basal lamina has collagen fibers while the reticular lamina does not have collagen fibers. They have elastic. They have elastic fibers made by the deeper connective tissue, consists of reticular fibers and ground substance. So the apical surface is always the free surface. So on your skin, the apical surface is the surface facing air. And inside your cavity, the apical surface is the one facing the inside, one facing the acid, one facing your intestine. So the basal surface is attached to the basal lamina. Okay, classification of the epithelia. So the number of the cell layers and the shape of the cell. So the classification is by cell layer. There's simple epithelia consists of a single layer of cells. So the simple epithelia is consisted of a single layer of cell All of them are attached to your basement membrane with its two parts, your basal lamina and your reticular lamina. Then there is stratified epithelia, where the cells consist of more than one cell layer. So what's the difference between I need this kind of thing. Okay, so when we want to compare simple epithelium to stratified. This is our basement membrane. You see the basement membrane? We said there's two parts of the basement membrane. What are the two parts? You have your basal lamina, and the second one is the reticular lamina, and below is your connective tissue. Okay, and up here is your epithelium. Now let's look at the difference. Epithelial, simple epithelium, all of the cells are attached to the basement membrane. All of the cells are attached to the basement membrane. But in this is simple epithelium. In stratified epithelium, stratified epithelium, what do you have? You have two types of cells. You have cells that, yes, they are touching the basement membrane like this. And you have other cells that are on top, on the head of the cells who are touching your basement membrane. These are stratified epithelium. So if you look here, simple epithelium, all of them are touching the basement membrane. Like in an Uber, okay, or in a van, everybody has a seat. They're sitting. But in the bus, some people are sitting and some people are standing up. Some people are on top of each other, for example, on top of each other's head. So that's what happens in the difference between stratified and simple epithelium. There we go. That's simple epithelium. They're all touching the basement membrane. But stratified, you see one layer touching your basal lamina. You see it right here. But the other ones, they are on top of each other. On top of each other. There is actually a third layer that they don't have in this book. It's called the pseudo-stratified. Pseudo-stratified epithelium. Pseudo-stratified epithelium. In pseudo-stratified epithelium, what happens? Let's say this is the basal lamina. This is our basement membrane. That's the basal lamina. That's the first layer of our basement membrane. Who is the second layer? Reticular lamina. Okay. In the pseudostratified, it's a mixture of both. So one cell has a part of it, and the other cells, some cells are touching, and some cells have a part of them that is touching the basement membrane, and the other part that is touching some other cell. So they look like that. So here, cell number one, you see part of it is touching the basal lamina, but the other part is touching cell number two. We did not see this here. Here, you're either touching the basement membrane, like these guys, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and some of them are touching other cells. But you don't have one cell who's doing both. That is what happens here in the pseudo-stratified. It's a gray area where they touch both. They touch the basement membrane and they touch the nearby cells. Okay. Classified by cell shape. So there's also another classification. There is squamous cells. Squamous cells are a sunny side egg up. Like you see, like an egg sunny side up, or a sunny side egg up, or egg sunny side up. Sunny side up. Sunny side up egg. No, there's no egg. It's sunny side up. Sunny side up. Okay. So you see the squamous cell is the sunny side up, and the yolk is the nucleus. So it's sunny side up. Cuboidal they have like a cube. Their length and width are the same. But columnar, they are more long, tall than they are wide. So the squamous cells, so there are squamous cells, sunny side up, and they are flattened, flat. And cuboidal cells, I'm going to get egg sandwich after this. The cuboidal cells are short cells, and the columnar cells are tall cells. Columnar cells are tall cells. Okay. Well, now I'm going to ask you guys, what's a squamous cell? Flattened. What's a cuboidal cell? No, no, no. It's right there. It's visualization more than remembering. That is the columnar. So now you know the short cell is like is like short person. They are short, meaning the length and the width are similar. So they are short. But tall, their length is more than their width.
So for the keyboard cells, they are all the same length, like they're all the same like size and everything.
Yeah,
Column.
columnar, yeah.
Like
Not everything, just the type. Yeah, so the columnar are longer than they are wide. So that's why when we call a tissue cell, we don't say it's stratified. We don't say it's stratified or simple. We say simple and what? Simple and squamous, simple and cuboidal, simple and columnar, or stratified squamous, stratified cuboidal, stratified columnar. If you say this too, that's half a point because now you're describing just the layer. Is it stratified or simple? But now you forgot to describe the shape. So you say simple can be one of three. Simple epithelium can be squamous, simple epithelium. Cuboidal, simple epithelium. Columnar, simple epithelium. Stratified, simple epithelium. Stratified squamous. Stratified cuboidal. Stratified columnar. So you see, simple squamous epithelium. Simple squamous is like this. Simple squamous, sunny side up. Okay, simple epithelia. So we have simple, obviously we said that. Simple squamous epithelia, simple cuboidal epithelia, simple columnar epithelia. And for stratified, we mix and match too. Stratified squamous epithelium, stratified cuboidal epithelium, and stratified columnar epithelium. So the simple squamous epithelium is a single layer of flat cells. Single layer of flat cells where oxygen, carbon dioxide, fluids, and ions to diffuse across. So the single layer of a flat cell and has oxygen, carbon dioxide, fluids, and ions to diffuse across quickly. So examples are in the lungs. So where do we find simple squamous epithelium? That would be in the lungs, in the kidney tubules, and lining the blood vessels. Lining the blood vessels. Okay. Then we have simple squamous epithelium. So that's the simple squamous epithelium. We find our simple squamous epithelium in the lungs, the kidney tubules, and lining the blood vessels. This is the simple squamous epithelium. Then simple cuboidal epithelium is a single layer of short square-shaped cells. The substances diffuse across rapidly, and they are found in the kidney tubules, respiratory passages, and the ducts of the glands and the thyroid gland. So this is a single layer of cube-shaped cells. That's simple cuboidal epithelium, and that is a simple. You see the simple cuboidal epithelium, and it makes sense that they are part of a gland. The simple columnar. Simple columnar epithelium or single layer of tall rectangular cells, and they are often with microvilli, as in small intestine and the kidney tubules, and often with cilia, as in the uterine tubes and some respiratory passages. That is where we find the simple columnar epithelium. Now there are, yeah, now that we're talking about this is the specialized epithelia, as the pseudo stratified columnar. So now the pseudo stratified columnar epithelium, they are simple epithelium that appears stratified, and some cells are shorter than the others, and the nuclei are at different heights. All the cells rest on the basal lamina, and they are ciliated in the upper respiratory passages and the nasal cavity, and the thickness provides a protective function. So you see the pseudostratified columnar epithelium. That's an example of pseudostratified columnar epithelium. And the pseudostratified epithelium, and this is your basement membrane, and that's the underlying tissue. Okay, transport across the simple epithelia. So there is a paracellular or transcellular transport. Somebody, I think, asked me last week transcytosis. What is transcytosis? Transcytosis is transcellular through the cells. So when the people pass between these train cars, you know, you can get a ticket for that. Okay, I didn't know that one, but yeah, I saw it. Going across the train will get you a $100 something ticket. Anyways, transport across the simple epithelium. So paracellular or transcellular transport. Paracellular transport, you would have substances that leak between the cells of the epithelial membrane. So most epithelial cells have tight junctions, making them impermeable. They have tight junctions, making them impermeable. So this is a paracellular transport where substance passes through the narrow space between the cells. You see, that's paracellular. That's paracellular transport. While transcellular transport is when the substance enters the cell via the active or passive means. And this is the substance that diffuses through the cytosol. See the substance diffusing through the cytosol. And the substance exits the other surface of the cell via active or passive means. Transcellular transport. In transcellular transport, the transport is by most epithelial cells. You would have in this transcellular transport, this is the way by which most of epithelial cells transport their substance. So the substance enters the cell through the phospholipid bilayer, and the substance diffuses through the cytosol, and it exits through the surface of the basal layer. So the paracellular transport, you see, the substance would pass through the narrow space between the cells, and the substance enters the cell via active or passive means, and the substance would diffuse through the cytosol, and the substance would then exit the other surface of the cell via active or passive means. Okay, summary of simple epithelia. So this is your simple squamous epithelium. That's the simple cuboidal epithelium. And this is a simple columnar epithelium. The pseudostratified, these are pseudostratified columnar epithelium. Components of the simple squamous are, you see that the single layer of cells that is flattened, and it provides a barrier through which gases and fluids can be exchanged, and it produces serous fluid, produces a serous fluid. The location are the air sacs of the lungs, and they are found in the air sacs of the lungs. This is the simple squamous, and the lining and the inner lining of the ventral body cavities and the blood vessels and certain parts of the kidney. That's simple squamous. And then there is simple cuboidal epithelium. In simple cuboidal epithelium, you have single layer of cube-shaped cells and round that have a round central nuclei, and there's absorption absorption, secretion of the mucus and other substances. And you see the kidney tubules that have found in the kidney tubules. These are the simple cuboidal epithelium. I'm not going to ask you at all on the exam one day, the cells that are part of the kidney, are they simple squamous or simple cuboidal or simple columnar or pseudostratified? I'm not going to do that. But this is just for you to appreciate what kind of cells are lining which types of organ to support that type of function. But the ones that are high yield are these unique pseudostratified epithelium. They have a single layer of columnar cells of different heights, giving a stratified appearance. And it secretes the mucus and propels it with ciliary motion. The nasal cavity, this is part of the male urethra and the upper respiratory passages. So this is very important to know. Where do we find the pseudostratified? In the nasal cavity, male urethra, and the upper respiratory passage, and they secrete mucus. You gotta know this one. This very specialized one. The pseudostratified columnar. Where do we find it? In the respiratory tract. All this mucus that's coming out. And the nasal cavity and the urethra of the male, not the female. Stratified epithelia. Yes.
Um,
Yeah. Yeah, it's posted. I posted it to you. Always I post it for you in the announcement section. Okay, I don't like to put it in the content, just in the announcement, so you can follow step by step, follow step by step. So if you just go through the announcement, you'll find it. You don't see it? Okay. All right. So can you guys just follow me here and just take your notes, and then I will give you. You guys like to have two screens, looking here and there. Yes, yes, that one. This is big enough. You can see that one. Okay. Stratified epithelia. And I'm also curious, and I will never think I will find a solution. This is all the colleges. The slides are here, the slides are there, and they still take photo. I don't know why this happens, but one day I will find out. Okay, yes. Okay, go use the restroom. Go use the restroom. Okay, give me that. Okay. Because you went to the other lab, I'm gonna appreciate that, you took extra time and you went to the other lab. So I really appreciate that. Everybody who went to the other lab, and now I'm telling you that bone practical, you got it this whole week. This whole week you are doing chapter 4, 5, and 6 of your lab manual, going to that room. I know what happens in that room. You gotta go to that room. You got to go in that room and you got to know your posterior from your anterior. You got to know your fossas. You got the learning center room. The learning center, you know it. That room. You got to go there. This whole week, the lab is coming and is a big exam and it's very hard. Yeah, yeah. Yeah, on the next lab. Please make sure you invest a lot. And this lab, yeah, it's the upcoming lab. The exam is the upcoming lab.
The bone exam?
Yeah.
Is this Friday?
Yes, yes. What do you think? Is the next one, yeah? The next lab 7.
Is it
No, it's identical. What were we saying? What were you guys last week?
So you're just gonna put a bone and then you say that's Friday?
I'm not me. They're gonna put it too. That's the thing. Otherwise, I was gonna give you a study guide. Everything them. This one, you gotta be very careful this week.
And then Sunday we also have a quiz.
Sunday, yeah.
Sunday's quiz is for what?
Sunday's quiz is for, where were you when I was saying this?
I was absent that.
No, I said this today after when we started the lecture. And I posted for you in the announcement. The quiz is gonna be after. Okay, we will talk about that after the lecture. Yeah. So the stratified squamous epithelium is keratinized. So it's divided into keratinized and non-keratinized. Excuse me? Keratinized means that it is rich in good, very good. So keratin is important because it gives that tensile integrity, it gives that strength, it gives that support And this strength and this support is what enables your own integumentary system to handle all of this temperature in the summer, all of this cold in the winter. And it makes the epithelium tough and resistant to friction, and it forms outer layer of the skin. So non-keratinized stratified squamous epithelium. So the apical cells are nucleated and they are living and they are found in organs requiring protection from abrasion but need to retain a moist surface. So examples are mouth, throat, the esophagus, the anus, and the vagina. Okay, so non-keratinized stratified squamous epithelium from the vagina. That's an example of the non-keratinized stratified epithelium. So we did simple cuboidal, now it's stratified cuboidal. So stratified cuboidal epithelium is rarer in the human body and it consists of two layers of the cuboidal cells. They line the ducts of the sweat glands. So there's stratified cuboidal epithelium from the duct of a sweat gland. So the stratified columnar epithelium is relatively rare in the human body and they appear columnar in apical layers and cuboidal in basal layers. So they line the ducts of the salivary glands, part of the male urethra, and the cornea. So stratified columnar epithelium from the duct of a salivary gland. Okay. The unique stratified epithelia. These are transitional epithelia named due to believe that the cells transitioned between the cuboidal and the squamous cells. So the transitional epithelia is only in the urinary system. They line the interior of the kidney, the ureters, the urinary bladder, and the urethra. The basal layers are dome-shaped and when relaxed, and they are flattened if they are stretched. So these are the transitional epithelium from the urinary bladder. So this is the summary of the stratified epithelium, whether columnar, cuboidal, and squamous. Now, glandular epithelia, we know that the tissue that is specialized in secreting substance is your epithelial tissue. So it grows inward into underlying connective tissue. And the secretory cells synthesize and release the products. So the classification of the glands is how they release their products and their shape. So some glands are exocrine. So exocrine glands secrete their content into a duct, while endocrine glands secrete their content into the bloodstream. Like for example, insulin, insulin and glucagon, they are secreted in a duct. They are secreted into the blood without a duct by the pancreas. That's why we call the pancreas a two-in-one. The pancreas is an exocrine and endocrine organ. The pancreas is endocrine because it secretes its hormones into the blood. It secretes insulin, it secretes glucagon into the blood. But it is also exocrine gland because it secretes trypsin and chymotrypsin into the intestine. Unicellular glands. Unicellular glands are one cell glands. These are like the goblet cell um, that secrete mucus and most common found in the digestive and the respiratory tract. Please remember this goblet cell. This goblet cell is important and it's found in the digestive and the respiratory tracts. So they secrete mucus protecting the epithelium. See, these are the goblet cell. They look like a goblet. You see, these are the mucus droplets. Multicellular glands. They are divided into simple, non-branching, compound branching glands, and these are your multicellular glands, simple glands and compound glands. The exocrine glands, we said that these exocrine glands, they secrete into a duct. So there are metacrine glands and holocrine glands. So the metacrine glands, their secretion is most common, and the salivary and the sweat glands, this is an example of the metacrine glands, which are the most common types of exocrine glands. They are found in your salivary and sweat glands, and the product is being released by exocytosis. And these are your secretory vesicles that are trying to make their way to the apical side. And these are your secretory epithelia. The packages product into secretory vesicles and are released by exocytosis. Holocrine secretion is different from mitocrine secretion in the sense that cells accumulate products and release them when the cell ruptures. So here is not exocytosis. Here is rupture of the cell. So the cells shed with products and they are replaced by new cells. So example are sebaceous glands. Sebaceous glands are very important because they shed and they rupture their content. They ruptured epithelia they rupture the epithelia, they rupture their content into the apical side. And the products accumulate in the secretory epithelial cell, and the epithelial cell would undergo mitosis to replace the shed cell. Okay, so now we have finished lecture number 12. So now let us go over um, lecture 13. So lecture 13 now is when we take our connective tissues. So we finished talking about our epithelial tissue. Now we're going to talk about our connective tissue. We said we have how many types of tissues? Four. We have muscular tissue, nervous tissue. Always have those two together. Muscular tissue, nervous tissue, and then connective tissue and epithelial tissue. Excellent. Two of those are excitable, while the other two are not excitable. Okay, connective tissues. Connective tissues are diverse with many functions that include connecting and building, support, protection, and transport. Support, protection, and transport. So the connective tissues are diverse with many functions that include connecting and binding. That's why they call it connective tissue. And that's why connective tissue is always found sandwich between the cells. Includes connecting and binding, the support and protection and transport. So connecting and binding. So they connect the structures in the body and they anchor the organs to one another and they bind other tissue layers together. They bind other tissue layers together. So the support, they are important not only for connecting tissue together and binding them together, they are important because they are there for structural support. So bone tissue type of epithelial, connective, muscular, or nervous? Bone. That's connected. How do we know it's connected? Because obviously bone is not excitable, so it can never be muscular, it can never be nervous. It can associate with them, but it is not nervous, it is not excitable, it is not muscular. Then it could either be epithelial or connective. Is the bone tissue stratified epithelial? Is the bone tissue secreting? Because remember, in order to be epithelial, you have to be able to secrete into gland. Does the bone secrete a form of a hormone or a form of a chemical? No, it's not. So definitely it is not epithelial. So that makes bone tissue by process of elimination, it makes it connective tissue. So the bone and the cartilage support like a steel girder, like a scaffold. It holds your body from collapsing and falling apart. The bone protects the internal organs. You know, when you have money, you put it in a safe. The safe from outside is made of iron. It's very strong. What about your brain? That's worth more than iron. That's worth more than money. It's worth a lot more than gold and silver, so it's protected by the skull. It's protected by the calvarium. Your heart is and lungs are protected by your rib cage. So the immune system components are present in the connective tissue. So for transport, the blood is a fluid connective tissue and it is the main transport medium of the body. The blood is a connective tissue and is the main transport medium of the body. The connective tissues are made up of cells and extracellular membrane, and the cells are surrounded by protein fibers embedded in ground substance. The extracellular matrix plays greater role in function. So the classification of the connective tissue. There are two types. There is a general connective tissue proper and specialized. General connective tissue proper and specialized. So the cells of the connective tissue proper are both resident and migrant cells. So the cells of the connective tissue proper are the fibroblasts, the adipocytes, the mast cells, the phagocytes, and the other immune cells. These are the fibroblasts, the adipocytes, the mast cells, and the phagocytes. Fibroblasts. These fibroblasts, remember, these are the cells of the connective tissue proper. Remember that the connective tissue is classified into general connective tissue proper and specialized connective tissue. It is made up of cells and the extracellular matrix, and the extracellular matrix plays a greater role in function, and the cells surrounded by the protein fibers are embedded in ground substance. Okay, cells of the connective tissue proper. These both resident and migrant cells. So the cells of the connective tissue proper are fibroblasts, adipocytes, mast cells, phagocytes, and other immune cells. These are the cells of the connective tissue proper. Fibroblasts. We said that fibroblasts are important for scar tissue formation. These are the most common types of the connective tissue proper. They are mature cells with properties of immature cells. They produce protein fibers and ground substance. They produce protein fibers and ground substance. Adipocytes are fat cells. Adipocytes are fat cells. Adipocytes are okay. Jane, can you please sit over there? Next week you come late or anybody comes late, no admission is zero. Okay? This is the last chance. Just take a pencil, please. Write your first, last name, and you have half an hour to sit on that corner, please. Thank you. Okay, so the adipocytes. So this is high yield. You gotta know what the adipocytes are. Adipocytes are the fat tissue cells. These are the cells that store. Remember the triglyceride and the glycerol? That is where they are stored, and they are butter, yellowish in color. And these adipocytes are a type of your connective tissue proper. They produce protein fibers and ground substance. So the adipocytes are fat cells. They are common resident cells and they contain lipid inclusions, and the nuclei are squashed to the side of the cells. You see the nuclei are squashed to the sides of the cells. These are your adipocytes. The mast cells. The mast cells are the largest resident connective tissue cell. These are the largest resident connective tissue cells. And the mast cells are the immune cells that secrete inflammatory mediators such as the histamines. These are the mast cells. These are resident sentinel mast cells. The immune cells secrete inflammatory mediators such as histamine. Phagocytes. Phagocytes are macrophages that may be resident or migrant cells in connective tissue. So you have neutrophils that are migrant immune cells. These are neutrophils that are migrant immune cells. Other immune cells. These are various cells that migrate in and out of the different connective tissue. What are the types of the connective tissue proper? Loose, dense, reticular, and adipose connective tissue. The loose connective tissue are also called areolar connective tissue.
Yeah.
All three protein fibers, the fibroblast and the other cells, are found in your loose connective tissue. It is found deep to the epithelium of the skin, the membranes lining the body cavities, and the layers of the hollow organs. So superficial skin, the epithelium, and this is the deeper layer of the skin. These are the fibroblasts. The fibroblast, that's the ground substance, and this is the ground substance. These are your protein fibers, the elastic fibers, and the collagen fibers, and that shows us the loose connective tissue. Then there is dense, irregular connective tissue proper, and that is made up of randomly oriented collagen fibers. It provides strength and resists tension in three planes, and it is found in the dermis, around the organs, and the joints. These are dense, irregular connective tissue from the thick skin. Okay. I'm sorry?
Is that like what's attendant to everything?
Yeah, so in order for the ligaments to be able to attach a bone to a bone, okay, that one we have to know that the ligament attaches bone to a bone and the tendon attaches bone to a muscle. In order to have that tensile integrity, it must have really dense and thick, irregular connective tissue. Okay? So the dense regular collagenous connective tissue is made up of parallel connective fibers, and it's strong since it resists the tension in one plane, and it is found in the tendons and the ligaments. You see, because it's very dense and very thick, very strong, we need that to hold this steel girder, hold this bone structure. So you see the dense regular collagenous connective tissue fiber from the tendon. So the dense regular elastic connective tissue, you have a parallel elastic fibers with randomly oriented collagen fibers. It allows the organs to stretch, and it is found in large blood vessels and ligaments in the spine. So the dense regular elastic connective tissue from the aorta, because a lot of blood is flowing through aorta with very high pressure. So we need the aorta to be stabilized with very strong, dense connective tissue to accommodate all of this blood that is pumped by the ventricle in such aggregate speed. So in the reticular tissue, you have reticular fibers and it supports the blood and the lymphatic vessels. It traps old and foreign cells in lymph nodes and the spleen. They form the basement membrane of the epithelia, the liver, and the bone marrow. Okay, so that shows you your reticular tissue from the lymph node, from the spleen. So that tissue, we find it supporting the blood and the lymphatic vessels, as in the spleen, as in the lymph node. It traps old and foreign cells in the lymph nodes and the spleen, and they form the basement membrane of the epithelia, the liver, and the bone marrow. So the reticular tissue from a lymph node. That is the reticular tissue of the lymph node. Adipose or fat tissue, adipocytes, is consisting of adipocytes, and there are few fibroblasts and little extracellular matrix visible. It insulate warms, absorbs and shocks and protects the major energy reserve. So adipose tissue from the hypodermis is in the superficial fascia. So the summary of the connective tissue proper, that is the non-specialized tissue, you have your loose connective tissue and dense connective tissue. The loose connective tissue is made up of fibroblasts, ground substance, and protein fibers. And the function of the loose connective tissue is to support, protect, and house the blood vessels supplying the epithelium. So that is always found in your basal lamina, and they are deep to the epidermis on the walls of the hollow organs and the membranes lining the body cavities. But the dense connective tissue is divided into three parts. There is irregular dense connective tissue, there is regular collagenous dense connective tissue, and there is regular elastic dense connective tissue. So the fibroblasts, the collagen fibers, and the ground substance, this is an example of irregular tissue. The regular collagenous tissue is the fibroblast, the collagen fibers, and the ground substance. And the regular elastic are found in your aorta. It's made up of elastic fibers. From the name, regular elastic. The reticular connective tissue is reticular fibers and leukocytes, and they form internal structure of many organs, lymph nodes, the spleen, the bone marrow, and the liver, and the basement membrane around the vessels and the nerves. The adipocytes connective tissue is made up of adipocyte and protein fibers. It's important for the warmth, insulation, shock absorption, and the protection and the major energy. It is the major energy reserve in the body. It is deep to the skin in characteristic areas, and they surround the heart and the abdominal organs. Now we finished with the connective tissue proper. Let's move into the specialized connective tissue. In specialized connective tissue, you have cartilage, bone, and blood. So this is not generalized tissue proper. This is specialized tissue proper. The specialized tissue proper is your cartilage that is making your ligaments and your tendons, connecting the bone to bone and connecting the bone to the muscle. And your bone tissue itself, comprised of the osteocytes and the osteogenic cells, the osteoblasts, who induce bone formation, and the osteoclasts, who degrade bone, and the blood tissue that is supplying oxygen and nutrients to tissue for oxidative phosphorylation. Let's begin with cartilage. Cartilage is tough, but it is not as tough as bone. Cartilage is tough but flexible tissue and is resistant to tension and compression and shearing forces. You just have to remember that cartilage is tough. It's very strong and tough, but it is not as tough as the bone because it is rich in what kind of proteins? Elastic and tensile proteins. Cartilage. Cartilage contains collagen and elastic fibers, and it contains chondroblasts and chondrocytes, and they contain avascular. They are avascular, and these are three types: hyaline, fibrocartilage, and elastin. Hyaline cartilage. Hyaline cartilage is the common, most common with more ground substance and fine collagen fibers. It ends of long bones forming the joints the ribs to sternum, parts of the respiratory tract, and the nose. And it is in most of the fetal skeleton. That is the hyaline cartilage. You have the trachea, and you have the hyaline cartilage from the trachea. Okay. There are there is also fibrocartilage. There's also fibrocartilage. Fibrocartilage is collagen fibers with less elastic fibers and little ground substance. These fibroblasts dump a lot of scar tissue. So when we are injured, we have scar tissue injury. We dump a lot of fibroblasts, and the fibroblasts secrete, secrete a lot of fiber, TGF beta. This fiber is going to dump on the injury site and elicit healing by fibrosis. There are intervertebral discs, and it is found in the intervertebral disc and the pubic symphysis. This is the intervertebral disc, and these are fibrocartilage from the intervertebral disc, and these are chondrocytes in the lacunae, and these are collagen bundles. Elastic cartilage. Elastic cartilage is made up of elastic fibers, and these are external ear and parts of the larynx, the voice box, external ear and parts of the larynx. And there's elastic cartilage known as the found in the external ear and parts of the larynx, and it is rich in elastic fibers. Now, when we talk about bone tissue is also known as osseous tissue. Osseous tissue. And ossification is the process of bone buildup, is the process of building up the bone. The bone tissue is the extracellular matrix has organic and inorganic components, organic and inorganic components. There are osteocytes in the lacunae, and there is a calcified extracellular matrix. So this here shows us bone tissue that is prepared so that the calcified extracellular matrix is visible. This bone tissue is important because not only it provides the tensile strength and protection of the viscera, these really soft, important organs, your heart, your brain, but it's also the bone is important for mineral homeostasis. When we have too little calcium, where do we get calcium from? We go and resorb the bone. We activate osteoclasts, and the osteoclasts will degrade the bone, and a lot of bone powder will be liberated into the blood tissue. And that will restore from hypocalcemia into eucalcemia. Then we have the opposite, and that is when we have too much calcium now. We don't want too much calcium because too much calcium can cause arrhythmia, can interfere with the depolarization, the action potential of the heart, the electrical stimulation of the heart. So too much calcium would cause premature contractions due to rapid excessive cycles of contractions. So that is very dangerous. And also too much calcium is going to lead to activating excessive enzymes that are supposed to be inactive. So people who have hypercalcemia always end up with some degradative lysosomal proteolytic metalloproteinase enzyme that was supposedly inactive, but due to the hypercalcemia, calcium is like an ignition. It turns on this off enzyme. So this off enzyme, due to the too much excessive calcium, is going to get turned on, and that's going to lead to degradation of pancreatic tissue. So there are three types of cells. The osteoblasts, these are the cells that, are you done with the student? Are you done? Can you please hand it in first, please, before you go? You have your first and last name in pencil. Okay. The osteoblast, the osteocytes, and the osteoclast. Osteoblasts, the osteocytes, and the osteoclast. So the osteoblasts, from the name blast, build up. They build up the bone, osteoblast. While osteoclasts, they cut up the bone, so they resorb the bone. So when I have too little calcium, which am I going to use? The ones who build up the bone or the ones who break the bone? Break the bone because I have too little calcium. I break the mountain of bone, and powder will come out, and then that will increase my calcium. But if I have too much calcium, is the osteoblast. I'm gonna take from the blood and dump it on the bone to have like mud, to have it like a really increased concentration. And then there are also osteoclasts. Osteoclasts. These osteoclasts degrade the bone tissue and I'm sorry, these are osteoclasts degrade the tissue. Osteoblasts build up the tissue. Osteocytes, they are like the computer. They are the ones who realize, is there too much calcium in the blood, or is there too little calcium in the blood? Do I need to start up osteoblast, or do I need to start up osteoclast, and so on? Okay? Blood. Blood is the connective tissue with fluid extracellular matrix. Blood is the connective tissue with fluid extracellular matrix. So the extracellular matrix is plasma composed of H2O, solutes, and proteins. The extracellular matrix is plasma composed of H2O, solutes, and proteins. There are formed elements that include the erythrocytes, the leukocytes, and the platelets. Formed elements include erythrocytes, leukocytes, and platelets. Okay, so the erythrocytes, these are the red blood cells, the leukocytes are the white blood cells, and the platelets. These are the platelets. So we know the erythrocytes are your. So this bone, if you cut the bone in half, obviously, if you look at a chicken bone that you put in a fried chicken, if you open inside the bone, it's gonna look black because it's fried with oil. But if you look at an original one or a bone of a cadaver that you slice it in half, it's gonna look white from outside, but when you open it, look in the inside. What color is that? Pink. Pink. That's it. Pink. No other color. No other color. No other color? Yellow. Yellow. Very good. Why yellow? The lipid. Very good. How did you know that? Wow. Very good. Excellent. So your bone, white from the outside due to the calcium. You cut it in half. It's red from inside due to the red bone marrow making these blood cells. And it's also yellow because there's lipid in there. There is butter in there. And this butter, this lipid is going to cause energy. It's going because the bone tissue needs energy. So keep watching these videos. See that pays. If you are watching some other nonsense, it's not going to pay you. You're going to waste your time. But this now pays you a lot because now you're going to get extra credit. That was very good. I'm very impressed. So utilize your time to your benefit. Try to fall in love with what you want to do in life. Okay, if you love education, you give it your all, it'll pay you back. Okay, make your education your hobby. Don't make it look like it's something you just gotta do. You have no other way of doing it. You just gotta do it. No, enjoy it, like it. If you invest more in it, you will become good at it, then you will like it. But if you don't dedicate a lot of time to your education, you're not going to be good at it. You're going to hate it. It's going to become a source of torture for you. So just invest more time. Invest more time. So the blood is made in your bone tissue, hematopoiesis. And the extracellular matrix is plasma composed of water, solutes, and proteins. And the formed elements include erythrocytes, leukocytes, and platelets. You see, these are the blood cells, and these are the leukocytes, and these are the erythrocytes. So summary of the specialized connective tissue. So that is cartilage. This is your cartilage, your bone tissue, and your blood tissue. The cartilage is made up of hyaline cartilage, fibrocartilage, and elastic cartilage. So hyaline is chondrocyte. These are your chondrocytes. That's your extracellular matrix. And it support, protects, and resist compression between the bones. It is found between the bones in the joints, between the sternum and the ribs, in the nose and the respiratory tract. The fibrocartilage is made up of chondrocytes, collagen fibers, and extracellular matrix, which support, protect, and resist compression. This is found in the intervertebral disc. There is elastic cartilage that is involved in producing and detecting the sound. It is found in the ears and the epiglottis of the larynx. This is the bone. The bone is the osteoclast, the osteoblast, and the osteocytes. It support, protect, and provide the leverage for movement. And the bone, and this type of bone is found in the bone tissue, and blood is found in the bone. And these types of tissues, the, this bone tissue, the white blood cells and the red blood cells, they are important for transferring, like we said, hemoglobin and storing myoglobin of this oxygen, and the leukocytes are to defend the the body from infectious organisms. Now, muscle tissue is only specialized for contraction. So when the muscle tissue is excited, it contracts. It does not relay a signal like the nervous tissue. So the functions are in the walking, the breathing, and the beating of the heart, and the narrowing of the hollow organs. So the cells are called myocytes. So there are three types. There are skeletal, cardiac, and smooth cell. There are skeletal, cardiac, and smooth muscle cells. The skeletal muscle, where most cells attach to the skeleton, producing body movement. It is a voluntary. The skeletal muscle system is under voluntary control. You see, you move your skeletal muscles as you like. It is not like your heart. That is not under your control. It does not beat by your control. It does not, your blood vessels do not constrict or vasodilate based on your control. So there's long cylindrical multinucleated myocytes called the Cuney fibers. So skeletal muscle tissue. The skeletal muscle tissue, so you would have skeletal muscle fibers. This is your endomysium, and these are multiple nuclei, and these are striations. So the cardiac muscle tissue. The cardiac muscle tissue is striated muscle that is found in the heart. It is mononucleated cells called the cardiac myocytes. These are involuntary, and the cells are short, thick, and branched, and they contain intercalated disc. You see? You see the striations, the cardiac muscle cells, the intercalated disc, the endomysium, and the nuclei. Smooth muscle tissue is a spindle-shaped myocyte. It is hollow organs that it's made up of hollow. It is comprises the hollow organs. It excuse me.
No, I'm saying I was right when I said earlier the muscle tissue, it has the blood vessels, the eyes and everything. You said I was wrong.
We're talking about what kind of muscle tissue. So you mentioned the eye and the skin. I'm talking about smooth muscle. So the muscle, there's three types of muscle. There's cardiac muscle, there's skeletal muscle, and there's smooth muscle. So this is smooth muscle. The smooth muscles are found in the hollow organ, the blood vessel walls. These are okay. When I say eyes, we mean the blood vessels that are in the eyes, not the pericytes, not the eye itself. So when you said eyes, I thought you meant like the eye structure.
No.
No, so this is the blood vessel that like gives the retina the blood.
Mhm.
You know what I mean? These are but it's okay. The eyes, the skin, and the ducts of certain glands, and most of them have gap junctions linked to other smooth muscle cells, and these are under involuntary control. You see these smooth striations, these smooth muscle cells are found in your lining your viscera, and these are responsible for the vasodilation and vasoconstriction, regulating your cardiac output. Nervous tissue. Nervous tissue is most of the brain, the spinal cord, and the nerves, and there are two types in your nervous tissue. The two types in the nervous tissue are the neurons and the neuroglial cells. The neurons are the central parenchyma of your nervous system. And the neuroglial cells, these are the cells that are supportive to these are the cells that are supportive to the nervous tissue, to the nervous system. So the extracellular matrix is with few protein fibers and mostly ground substance. The neurons send and receive information through electrical signals. The neurons send and receive information through electrical signals. We talked about that, that when excitation happens, we mediate an input signal like a computer. We mediate integration of that signal. We generate an output of that signal through the membrane voltage changes, through action potential. So when the neuron gets excited, it undergoes this conformational change. This undergoes this mediation of this action potential. So the neurons send and receive the information through electrical signals. The neuroglial cells perform supportive functions. The neuroglial cells and the axon and the cell body of the neuron, the nucleolus, the nucleus, and this is your extracellular matrix. These are the dendrites. Dendrites are emanating from the terminal ends of your neuron. Now, what are the membranes of these nervous cells? So the thin sheet of one or more tissue lining the body surfaces of the cavity. So most of the membranes aligning the cavity, the hollow organs, protecting the viscera, these are epithelial tissues and they have layers of the basement membrane, the reticular lamina, the basal lamina. These are the layers that mediate the passage of blood and nutrients from the connective tissue into these organs. And many of them contain a thin layer of smooth muscles. What are the functions of the membranes? They anchor organs and they serve as barriers and they are important for immunity. They are providing a fence, a form of innate immunity, natural immunity, generalized immunity to prevent everybody who's a threat, whether the bacteria or the viruses, the parasites, the fungi, no matter who they are, they prevent them from entering into the tissue. And the membranes are obviously important part of the glands because the glands are secreting their substance either into the blood, being endocrine glands, or into a duct, being exocrine glands through the membranes. So the membranes may be true membranes or membrane-like structures. So true membranes do not open to outside of the body. Examples would be serous and synovial membranes. These do not open to the outside. These are on the inside, like in your synovial fluid in the joints and the serous fluids lining your trachea, lining your stomach. And there are membrane-like structures. These membrane-like structures open or lines external body surface, and examples are mucous and cutaneous membranes. True membranes, these are the serous and the synovial membranes. Whoever has that sound, can you please turn that off? So serous and synovial membranes. What are serous membranes? These are also known as the serosa. So these serous membranes are protective membranes. They have fluid in them. This fluid is serous fluid, electrolytes, solutes. These line the pleural and the pericardial and peritoneal body cavities. They consist of the mesothelium, so simple squamous epithelium, basement membrane, and loose connective tissue. You see, these are your mesothelium, followed immediately by your basement membrane. This is the basement membrane, mesothelium, basement membrane, and lastly, there is loose connective tissue. And these are the serous fluids. These are the serous fluids. The serous fluids are like a bag of liquid surrounding your lung, a bag of liquid surrounding your heart to protect it from damage. So these serous fluids are important because okay, so if this is my heart, okay, if this is the heart tissue, the serous fluid is like fluid here that is like a cushion protecting the heart while the heart is beating. And it's also like if this is our lung, there is a surrounding fluid, there is a surrounding fluid, and this surrounding fluid is going to this fluid that is surrounding the lungs is providing the lung with a form of a cushion that provides it from trauma and holds its integrity and participates in the pressure gradient, as we're going to learn when we study our respiratory and cardiovascular system, we see that if you make a hole, you puncture this serous fluid, it leaks out, then you disturb your heart from compressing. When you take a deep breath, air is not going to go all the way to your lung because that serous fluid was also part of regulating the amount of air that enters into your lung and into your and the blood entering into your heart chambers. So it's very important to see that this viscera is lined with your serous membranes. So the mesothelial cells produce serous fluid between two layers, and the fluid lubricates the organ, allowing the movement without friction. There are synovial membranes. These synovial membranes line the cavities surrounding freely movable objects, freely movable joints. And the same way the serous fluid lined, if you see the serous fluid lines the heart, the synovial lines the bones, protects the bones from the friction as the bones articulate with one another. There is two connective tissue layers. So the inner tissue layer has synoviocytes. These are fibroblasts secreting synovial fluid to lubricate the joint. These are synoviocytes secreting synovial fluid. These are fibroblasts secreting synovial fluid into to lubricate the joint. And there is external connective tissue with loose and dense irregular connective tissues. These are loose and dense connective tissues. So membrane-like structures, these are mucous and cutaneous membranes. The mucous membranes are also called the mucosa, and that is found obviously in your lung. They line the body passages opening to the outside of the body, and they contain goblet cells secreting mucus, and the mucus provides protection. So we find these mucous membranes lining the inside of our respiratory tract as well as the digestive tract. Cutaneous membrane, that's your skin. So when these tissues aggregate together, they make membranes. So the cutaneous membranes contain keratinized stratified squamous epithelium called the epidermis. That is cutaneous, simple epithelium. Now, we talked in the past about when the tissue is damaged. It is damaged either by spinning the cell cycle, entering the cell cycle, and birthing and bringing more cells. What is that known as? What is that known as?
What? Regeneration.
Regeneration, healing by regeneration when we are able to get more and more cells. Okay, and fibrosis is when we heal in a tissue that is not regenerative, like the neurons, the nerve tissue, that is not regenerative. Or maybe even a regenerative tissue like the skin, but the injury was so deep, it cut down like a burn or a laceration that went so deep, it cut the top layer, the epidermis, the layer below it, the dermis, the layer below that, the hypodermis. You remove even the stem cells that are able to cause this regeneration. The cells that are progenitor cells that have the potential of entering the cell cycle and dividing again. Those cells are gone. They are scraped out. They are removed. So how are you going to be able to have a cell cycle? You will not. So then even if it's a regenerative tissue, it is now will heal with healing by scar tissue formation, healing by fibrosis. So you have to understand that we have healing by fibrosis and healing by regeneration. We heal by regeneration if the injury is so light and on a tissue that has the capacity and the potential to regenerate and enter the cell cycle. But if the tissue does not have the capacity to enter the cell cycle, like heart tissue, like brain tissue, if that is damaged, then it's damaged for good. And we do not heal with regeneration, we heal with fiber tissue deposition. So cells known as TGF beta, these TGF beta cells will arrive to the site of the tissue and dump so much fiber, and this fiber is like cement, and you're gonna have scar tissue formation or healing by fibrosis. You see regeneration? So damaged cells are replaced with the same type of the cell. You see damaged cells are replaced by the same type of the cell. And the cells are replaced with new epithelial cells. The cells are, you see, that's regeneration. You see the cells are damaged, but other cells came on their place. Do you guys see? You guys see regeneration? The cells are damaged. Now they are healing by regeneration. Now let's look at fibrosis. Some tissues incapable of full regeneration. So the fibroblasts divide and use collagen to fix the injuries. So it results in scar tissue, a type of dense, irregular connective tissue. So the injured area, you see, now the injury went so deep that you cannot heal it. Or in you see the cardiac muscle tissue, it cannot regenerate. So it will heal by fibrosis. So you see, you got to understand the difference between tissue repair by regeneration and tissue repair by fibrosis. So the epithelial tissue, in regeneration, the healing happens in epithelial tissue, in connective tissue, except cartilage. So cartilage does not heal by regeneration. And smooth muscles, they are able to regenerate. That's why when you're injured, and you, for example, if somebody has physical abuse or trauma to their skin, and these blood vessels are ruptured, and you see like a bruise, after a while it goes away because your body would remove that clotted blood and that necrotic debris, and it can initiate the blood vessel cells to enter the cell cycle. So you're going to have angiogenesis. Genesis is making. Angio is blood vessel. So you're going to have new blood vessels sprouting of blood vessels, known as angiogenesis. I have videos about that. I'll post it for you guys to see it. So that is these smooth muscle cells are able to regenerate. I'll show you right now the let me show you. We are done with 13. Let me show you the so if you go online, okay. If you go online and you go to my YouTube channel and you go to videos, you can find a video of me talking about the different types of. Yeah, so this is, okay, so that is healing by regeneration and healing by, okay, so this is healing by, do you guys see regeneration? So that's the normal epithelium right here. You see the normal epithelium? Here it says the tissue harm leading to loss of structural and functional integrity of the tissue. That's the normal tissue. The normal tissue underwent this damage. You see the damage in the middle? You see the damage? Now I have two paths. 2B, that's step number one is the damage. Step one, that's step one, the damage. Step two is 2A or 2B. 2A is if the injury is reversible and limited injury, we heal with this green regeneration because the injury was so light, we go right back to normal tissue. But if the injury is irreversible injury, there will be inflammation. You see inflammation? See the macrophage coming? See the macrophage? And you see the inflammation and the T lymphocytes, they would come and then they would remove the damaged tissue. And then these yellow, you see the fibroblast? These yellow fibroblasts would come and dump here, and you would have healing by fibrosis. So you can watch this video at home. Okay. I have also another video here. Oh my God, I gotta do this every time. If you go down to here, that's tissue repair. So if you look at tissue repair, so look at this. Here you had normal tissue. Okay. There is a mild superficial injury like this one. You see, it's really mild. That's my normal tissue, right? And this is an injury only to the surface. Then the next step is a regeneration. You see regeneration. So everything regenerated. But look at the other route. When the severe injury, now the injury is not superficial like here. The injury crossed, it went deeper layers. It's not only in the epidermis no more, it's in the dermis and the hypodermis. Then you would have scar. You see the scar tissue formation. So that shows you here. If the injury is so deep, then these fibroblasts will be recruited and they are going to, if the injury is this is an example of injury that is superficial. So the injury is at the top, then you see the new blood vessels coming out. This is the blood vessels sprouting, known as angiogenesis. And you see we went right back to normal tissue. We don't have scar. We don't have scar deposition. So those videos can help you a lot. So you may want to watch those. Okay, so now it's time to do the last two lectures. So in order to So now we're going to go into the integumentary system. We've talked about the integumentary system. That's your skin. Your skin or the integument is what kind of tissue? Is it connective tissue? Is it muscular tissue? Is it nerve tissue? Or is it epithelial tissue?
Is it connected? No, it's connected?
Epithelial tissue. Epithelial tissue. So that's the skin structure. The skin structure is the skin is the largest organ with about 10 to 50% of the body weight. And is also called cutaneous membrane. It is composed of the epidermis, the dermis, the accessory structures, and the hypodermis, but not part of the skin. This is the epidermis, this is the dermis, and this is the hypodermis. That is not part of the skin. This is the hair, this is the sebaceous gland, and this is the sweat gland. This is the adipose tissue, and these are the blood vessels. These are the blood vessels. You see, you got to know your three layers. The topmost layer is your epidermis. The second layer is the epidermis. And the last one is the hypodermis. Where does the hair start off? The hair shaft come from the dermis, the middle layer. You see it's right here, the middle layer. And where do you have a lot of fat, yellow, adipose tissue? Hypodermis. Hypodermis. So the epidermis is a superficial and avascular. You see, it has no blood vessel. It's avascular. Who is vascular? The hypodermis and the dermis. It is composed of keratinized, stratified, squamous epithelium. Got a lot of keratin to protect us from this acid mantle that is secreted on our skin to protect us from this invading bacteria, invading microbe called acid mantle. I can see it right here. Acid mantle. I erased it. So it's pH 4 to 6, and it burns all of the bacteria, microbes, fungi that are trying to come in. And it is also important to protect us from the sunscreen, or from the cold, the hypothermia. So it is composed of keratinized stratified squamous epithelium. So the epidermis is the outer layer, and it consists of loose connective tissue and dense irregular connective tissue. Loose connective tissue and dense irregular connective tissue. That's the dermis. See, loose connective tissue and dense irregular connective tissue. The accessory structures are embedded in the epidermis and the dermis. They include sweat glands, sebaceous glands, hair, the nails, the sensory receptors, and the arrector pili muscles. So you see, this is the hair. Can you guys tell the hairs? And these are the sebaceous glands. These are the sweat glands. And these are your adipose tissue. These are the arrector pili muscle, and these are the sensory receptors. The hypodermis, also called superficial fascia or subcutaneous tissue. So the superficial fascia or the subcutaneous tissue. They are deep to the dermis, but not part of the skin. Deep to the dermis, but they are not part of the skin. They anchor the skin to the muscle and the bone. They contain loose connective and adipose tissue. They are highly vascularized. So it's very important to know that the hypodermis is the subcutaneous tissue, and has a lot of adipose fat tissue. You see, that's the adipose fat tissue, and these are the blood vessels. Functions of the integumentary system. So the integumentary system is important for protection, protecting us from sun, protecting us from cold, protecting us from invading microbes that are trying to make their way inside our body. The integument is very important for skin. It has a lot of sensitive receptors, tactile receptors. That's why when the mom touches the skin of the baby feels calm, and that gentle touch will calm the baby. And this is not just for the baby, for all the human beings. We have this, when you shake hands, you can feel that somebody's touching your right hand. If you're shaking with the right hand, not on the left, because the tactile receptors on your right hand have been stimulated, because their action potentials are open, because their action potentials are have been initiated due to the opening of the stretch sensitive membrane channels. So when you shake somebody's hand, their receptors would open, and you would have influx of uh, ions, and influx of other ions. Vitamin D synthesis. Okay. Protection. So the function of the integumentary system is to protect from mechanical damage. That is keratinized stratified squamous epithelium, and provides durable but flexible surface. And the protection, the immune system protection from pathogens. So it protects you from the risk of infections, and increases if the skin is cut, and it has antimicrobial secretions known as the acid mantle, known as dermacidins. Protection. So it's very important to see how the integumentary system is important for protection, because it protects from the environment. So the skin absorbs ultraviolet radiation. We talked about that. And there are lipid secretions that repel the molecules with polar, covalent, and ionic bonds. The sensation, you would have sensory receptors that allow the brain to perceive the changes. So there are hot, cold, and pain receptors. There's a thermoregulation, and thermoregulation helps to maintain constant internal body temperature. So we know that our skin is highly implicated in when it's too hot. What happens? Our red blood cells, our blood that is following into these blood vessels, and the blood vessels are flowing into the connective tissue beneath our basement membrane. That blood vessel diameter is going to increase to accommodate more blood carrying these heats from the core of your body to the periphery, to your skin, and now this heat is going to dissipate from your skin, from your body into the air, and now you will feel calm. Vasodilation? That's vasodilation, yeah. So we're gonna vasodilate the blood next to the skin. And when it's too cold, we're gonna vasoconstrict the blood vessel close to the skin. So that shows you how the skin is important in thermal regulation. And also when skeletal muscles, shivering thermogenesis. When the skeletal muscles are you're shivering, whose is your skin. So your skin is very important. Skin is also important for facial recognition. You know, there's like $1 billion industry on use this cream, that cream. This makes your skin look beautiful. That makes this whatever. People cannot leave the home without looking in the mirror. Okay, that's not me, but many people. And, and then you have some people who have like are very preoccupied with their social image, with their self-image. So, you know, that's why there's plastic surgery. So your skin has a lot to do, and Botox. Yeah. And you know, in as a nurse, you know, you're gonna be dealing with, you know, patients who come in, you have to read their face. And that is part of the mental assessment exam, a psychological exam to tell if the patient is suffering with some sort of anxiety or the patient suffering from depression. That could be told by the skin, by looking. Or if somebody's angry, they get reddish skin, you know, or so you can look at the skin and tell, even if the patient is nonverbal, autistic, you can tell their feeling just by looking at their face. But the veterinarian cannot look in the face of the cat or the monkey or the dog and tell if they have depression. It doesn't mean the animal doesn't have depression. Animals do have anxiety, they do have depression, but you cannot detect it with their skin because their body is not having the same type of architecture. Our skin is so intimately in touch with our nervous system and our skeletal muscle system. So we can be able to, you know, have a smile, we can have a some form of, you know, to show affection with your physical, with your facial features, but they cannot do that as animals. So the skin is very important. And skin is important for excretion. So we excrete, people think that excretion only happens through the digestive system. That is not true. Your integumentary system, the waste products are eliminated through the skin, and examples are lactic acid and urea. Vitamin D is important because vitamin D is synthesized on your skin. So the skin converts cholesterol into vitamin D, and uh, ultraviolet light converts the precursor into cholecalciferol that enters the bloodstream. So the liver modifies cholecalciferol, and the kidneys form calcitriol, which is a form, the active form of vitamin D. Then when we have vitamin D successfully made, what are we gonna end up with? Why is vitamin D important? Active vitamin D. This is inactive. We're trying to make active vitamin D. Why is that? What about it for the bones? Yeah, what does it do to the bones? Vitamin D makes the bone how? Excellent, excellent. It helps in the absorption of calcium from the intestine. It absorbs calcium from the intestine. Now you have the epidermis, that's the upper layer. So that's the superficial layer of the stratified squamous epithelium, and it is composed mostly of keratinocytes. So there are 95% of the epidermis cells. These are the keratinocytes, and they make, also known as corneocytes. They make protein keratin to make the skin tougher, and they are linked by desmosomes, making the epidermis stronger. Linked by desmosomes, making the epidermis stronger. So there's a sloughing of the keratinocytes, and there's a stratum corneum, there's a stratum lucidum, and there's a stratum granulosum. The stratum spinosum and stratum basale. Keratinocytes. Keratinocytes are arranged into layers of strata within the epidermis. There's the stratum basale, which is a stratum germinativum, and there's a stratum spinosum, and there's a stratum granulosum, stratum lucidum, and stratum corneum. These are the layers that are within the epidermis. The stratum basale, known as the stratum germinativum. This is the deepest layer. That's why they call it the base, stratum basale, because it's at the base. It is a single layer of stem cells on the basement membrane. It is the most metabolically active, producing vitamin D and the other epidermal layers. You see the sloughing. This is the sloughing of the keratinocytes or the dead cells. And at the basement layer, you have the stratum basale, then the stratum spinosum, then the stratum granulosum, stratum lucidum, and at the top, you'd have your stratum corneum. Usually, the high yield ones, you don't have to memorize the five, but you got to know the most superficial one where you have the sloughing of the cells, when you have the desquamation or the exfoliation of these dead cells, that layer is known as the stratum corneum. But at the base, you would have stratum basale. There's the stratum spinosum. Stratum spinosum is the thickest layer and superficial to the stratum basale. The cell artificially appear spiky and mitotically and metabolically active. They also produce vitamin D. Stratum granulosum, the middle layer of 3 to 5 rows of keratinocytes. They are named for prominent cytoplasmic granules. Uh, Keratin is one type of granule, and others are lipid-based substances that blocks the hydrophilic molecules. It maintains the internal fluid and electrolyte homeostasis. The stratum lucidum is only in thick skin of the palms and the feet, and superficial to the stratum granulosum, and it contains thin, clear layer of dead keratinocytes. So this is the middle. So we just need to know the one that is mostly deep, that is the stratum basale, and the one that is the most superficial, known as the stratum corneum. The stratum corneum is the most superficial layer of the epidermis. There's several layers of dead, flattened keratinocytes, and the cells lost their organelles, and they only contain keratin. So these cells only contain keratin, and they lost the desmosomes, and the cells shed, as we said, or exfoliate or desquamate. 1 million keratinocytes shed each day because they have lost their organelles. These are dead, flattened, superficial cells. These are sloughing cells, exfoliating cells, desquamating cells. Okay, so when we look at the epidermal growth, in the epidermal growth, 40 to 50 days from the stratum basale, from the basement, until they shed from the stratum corneum. So mitosis of the stratum basale pushes the cells outward in the epidermis. Mitosis of the stratum basale pushes the cells outward in the epidermis, pushes the cells outward into the epidermis. So we grow in an inverse fashion. Do you see this is our normal cell? Our original cell is undergoing mitosis. You see the mitosis? You see the two cells polarized to the opposite regions, and then they are going to divide. You see now they divide A divided into B. Then B is moving up until they die at the surface as keratinocytes. They lose their organelles and they die. So other cells of the epidermis are the dendritic Langerhans. So these dendritic Langerhans cells, they are like immune cells. These are cells that are resident sentinel cells like police precinct. They are there to reconnoiter. They are there to monitor who's about to enter into the tissue. And these dendritic cells, they themselves, they can fight all the bacteria, the parasites, the infectious microbes, but they can make a phone call to the blood to recruit the second line of defense. So we have the police, the cops, 911. You call them for generalized function, theft, littering, trespassing, assault. They come for general things. But then you have the second specialized ones. These are your detectives, your CIA, your FBI. You have a division for robbery, a division for homicide, a division for arson, all of that stuff. So your Langerhans, they are like the cops. They are general. They deal with any bad person. But the problem is, then they have the ability to contact those monocytes. These are the detectives, these B cells, these B cell secreting antibodies, these T cells able to perform cytotoxic damage. The dendritic Langerhans cells are Merkel cells and melanocytes. The dendritic Langerhans cells, they are found in the stratum spinosum, the phagocytes of the immune system, um, found in the stratum spinosum, phagocytes of the immune system. And you see, this is the slowing off of the keratinocytes. This is the stratum. This is the stratum lucidum. And there's the stratum granulosum. And there's the stratum spinosum. Merkel cells. Merkel cells are found in the stratum basale of the fingertips, the lips, and the base of the hairs. These are the touch receptors. These are the ones who can make you feel the sensation of touch. These are the Merkel cells. So on the exam, when you're asked the dendritic, they are phagocytes. These are the police precincts. But when you're asked the Merkel cells, these are the cells that relay the sensation of touch. They are mechanical receptors, stretch receptors. They are sensory receptors associated with neurons of the dermis. They detect light touch, differentiating shapes and textures. How you know that this is a really pointy end, and that's sometimes we do a neurological examination when the patient, we tell them to close their eyes, and then we hit them with a cotton and with a small pin and tell you, what did I touch you with? And then they can say, you have touched me with a pin, or you have touched me with a cotton piece. That means their Merkel cells are working. But if they have a stroke, and then that part of their brain is unable to receive information input from their Merkel cells, you touch them with a pin, they're like, I feel like something vague. You're touching me with something vague. That's why after anesthesia, you feel like, because sometimes you're gonna feel the touch really well because your Merkel, but you gotta know what Merkel cells are. That's for your touch receptor. Melanocytes. Melanocytes are the cells that secrete melanin. Melanin is like the sunglass, the sunscreen for the DNA. It's like when you put, the black garbage bag, you put it over the DNA because the DNA, what happens when ultraviolet radiation hits the DNA? What happens? What happens when, I'm sorry?
Skin cancer.
Yeah, you get a mutation. The mutation leads to skin cancer. You get damage. You get mutation damage. You get ultraviolet destroy the DNA. So you have this melanin to screen your body from this ultraviolet radiation. When I visited back home in Africa, is it very hot there? So when I come back here, people tell me your skin got a lot darker. And when I stay here for a while, my skin lights up a little bit than before. And the reason is because of the melanin. Over there, the sun was hitting me so much, my body started to make melanin in order to protect my DNA in the nucleus from being damaged. But here, not as much sun, so the adaptation would go down. We're not going to have as much melanin. And people, when you look at somebody who has a really dark skin complexion compared to somebody who does not have as much dark skin complexion, the reason is not because the person with a dark skin complexion has more melanocytes. No, we have equal number of melanocytes. But the person with a darker skin complexion, they secrete more melanin. So you could have 10 grocery stores in each neighborhood, but one sells $1 million a day. The other neighborhood they sell 500,000 a day. So that shows you the same number does not mean you're secreting the same amount. So the people who are darker in color, they secrete more melanin. They don't have more cells, they secrete more melanin. So these are the melanocytes. You see, they are found, the melanocytes are found in your dermis, and they are at the junction between your dermis and your epidermis, and they secrete melanin. There is thick skin. Thick skin is found in the palms and the soles. So it is found in areas of mechanical stress. So obviously, if you look at the eyelid, that's skin too, but it's so thin. If you look at the palm, that's thick. I cannot clap with my eyelid. It's gonna hurt my eye because my eye is so thin because that area is not undergoing a lot of stress. If you had a really thick eyelid, it's gonna like be so heavy on your eye. It's gonna cause trauma to your eye. And that's why the soles of the feet and the palms are really thick. There is no hair follicles, but numerous sweat glands. You see, this is the thick skin. The thick skin, the thin skin has a the thin skin has a thinner dermis, while the thick skin has a thicker dermis. With the thick skin contains all five epidermal layers and a very thick stratum corneum. No hair follicles, but numerous sweat glands. The dermis is deeper to the epidermis. It is highly vascularized, and the functions are blood supply for the epidermis. There are sensory receptors, and they anchor the epidermis. So the epidermis is anchored on the dermis, and the dermis is divided into papillary and reticular layers. The papillary layer is the superficial and thinner layer of the dermis. The papillary layer is a superficial and thinner layer of the dermis. So it is composed of loose connective tissue, and the collagen fibers anchor the epidermis in place. The collagen fibers anchor the epidermis in place, and the projections called the dermal papilla, and they contain capillaries providing nutrients to the epidermis. The projections called the dermal papilla, they contain capillaries providing nutrients to the epidermis. The papillary layer. The papillary layer has a tactile or mesonar corpuscle that is found in the dermal papilla, and these are sensory receptors for light touch. These are the greatest in the fingertips, the lips, the face, and the external genitalia. You see, these are the dermal papilla. They are right beneath your epidermis. There is a reticular layer of the dermis. This is the deeper and the thicker layer of the dermis. It is a dense, irregular connective tissue that is continuous with the papillary layer and the hypodermis, and it is strengthened by the collagen fibers. There are elastic fibers that allow the dermis to stretch. The reticular layer is known as the lamellated or Pacinian corpuscle. They respond to pressure and vibration. These are in the lenticular layer, you have your lamellated corpuscles. They respond to touch sensation. Skin markings. There are dermal ridges, epidermal ridges, and tension or cleavage lines. These dermal ridges, they form from dermal papilla in thick skin with thick collagen fibers. They indent the epidermis, producing epidermal ridges to enhance the gripping ability of the hands and the feet. You see, these are the dermal ridges. That's how we are able to have a really strong grip. These are the dermal ridges. These are the patterns unique for every individual. These are what make the fingerprint. So the sweat pores along the ridges leave a fingerprint. So these are tension and cleavage lines, and they are clinically important since the incisions are parallel to the tension lines. Okay. So we talked about melanin, and then melanin ranges from orange red to black, and the synthesis in the melanosomes of a melanocyte. The synthesis increases from the ultraviolet exposure to protect the DNA of keratinocytes. The melanin oxidizes and darkens, and the ultraviolet would cause damage to the melanin. We've talked about that. Keratin is very important because it gives us it interact with the sun, but it gives us in the end vitamin A. So keratin gives us vitamin A. Hemoglobin, that's why we see there's redness in the skin due to the blood flowing to that part of the skin. So the skin color is a diagnostic tool. We talked about that. If a patient is has a bruise, has physical abuse, cyanosis is when there's like a breaking of these blood vessels, or maybe there is not enough blood supply to the tissue and the tissue is suffocating, dying from necrosis. That is known as cyanosis. Jaundice is when there is a severe discoloration of skin color and sclera of the eye due to the breakdown products of hemoglobin. Due to the breakdown products of hemoglobin. So the yellow skin due to the liver dysfunction. Okay. The last thing we're gonna end with is your. So this is like a couple of few slides. We've talked about this, but we just gotta cover them real quickly. Then I will end the recording session, post it for you online if you need to go back. Then I will talk to you some more about this lab practical. The skin derivatives and the pathology. Okay, so when we study the anatomy and the physiology, we also have to understand the pathology of what goes wrong. So the structure and function of the epidermal derivatives, such as the hair, glands, and the nails. So lacerations, burns, and skin cancers are of a major concern. So we know that the hair functions in thermoregulation, protection, and it's also important for the sensation. So the pili of your hair, they are derived from the epidermis. They consist of keratinized, stratified squamous epithelial cells, and the amount of melanin determines the hair color. The amount of melanin that determines the hair color. So the shaft, this is the hair shaft. The hair shaft in the dermis, known as the dermal root, and the shaft has dead keratinocytes and above the skin surface, and the root is in the dermis and embedded in the hair follicle epithelial root sheath. So the hair structure, the root has sensory neurons called hair papilla. The root and the hair papilla is in the hair, you see the hair bulb, and the matrix is the area of the keratinocytes that would undergo mitosis. There are three regions in the transverse section. There's the cuticle, that's on the outside, the cortex, that's the middle layer, and the medulla, that is the core of the hair follicle. So the dermal root sheath supports the follicle. This is the dermal root sheath. And the arrector pili, smooth muscles, causes the piloerection, so the hairs stand up to produce goosebumps. So there's a stimulation of the arrector pili, and that what leads to the hairs to stand up. Alopecia. Alopecia is baldness that occurs in both sexes. That's thinning of the hair. The lanugo is thin, non-pigmented hair of the fetus that falls out at birth. The nails are derived from the epidermis. So the nail plate is the largest part. That's the nail plate. And this is the nail root. Is divided into the nail body that could be seen and the nail root that is lying underneath the skin. So the nail matrix part of the nail root is containing actively dividing parts. These are the actively dividing parts of the nail. So in the nail you have the eponychium or the cuticle. It consists of the stratum corneum. There is a hyponychium that is the distal end attaching to the nail bed. There is a lunula, which is an opaque region of keratin accumulation. That is the aponechium. Skin glands. Skin glands are sweat and sebaceous sebum or oil glands. The sudoriferous sweat glands, they are four types known as the eccrine. These are the most types that are common in the sudoriferous sweat glands. There are apocrine glands. These are in certain areas of the body. So there are ceruminous wax that is secreted by these sudoriferous glands, and there is mammary glands that produce the milk. The eccrine sweat glands, these are simple tubular glands in the dermis. The papillary, they are primarily important for thermoregulation, and the sweat exits the duct and leaves by a sweat pore. The sweat is 99% water plus electrolytes, lactic acid, and antimicrobial compounds. That is from your eccrine sweat glands. You see, these are your eccrine sweat glands that are bursting out all of their sweat from their core into the lumen of the duct. Apocrine sweat glands, these are found in the axilla, in the anal area, and the areola. These are thick, protein-rich sweat metabolized by the skin bacteria, and the secretion is through the hair follicles and regulated by skin hormones, and secretion begins at puberty. Ceruminous gland. Cerumen is secreted where? From where? Where do we find cerumen? Ceruminous gland. In the ear. That's the earwax. It secretes cerumen, earwax into the hair follicles and it lubricates the tympanic membrane, the eardrum, and traps the particle. The mammary glands. These are specialized sweat glands that produce milk. The milk contains proteins, lipids, sugars, and other substances. There are sebaceous glands. Sebaceous glands are branch glands with clusters of secretory acini cells with small ducts. Sebaceous glands, they secrete sebum, waxy oily mixture of various lipids. The ducts empty into the hair follicles or small pore. Most are found on the face and the scalp, but are absent on the palms and the soles. They use holocrine secretions in which the sebum accumulates until the cell ruptures. The sebum contains cell fragments and debris, and the secretion is influenced by hormones and increases after puberty. This is the sebaceous glands. These are the apocrine glands. Skin pathologies are your trauma to the skin, the lacerations, and the burns, the primary first-degree burn that is only on the epidermis. So the epidermis is the primary first-degree burn. Second-degree burn is when we scrape the epidermis and part of the dermis. And the third degree is when you remove the epidermis and the dermis and part of the hypodermis, and that is very dangerous. You're going to lose a lot of water, cause hypovolemia, you're going to undergo an infection. So the body is divided into 11 areas to estimate the percentage of affected body surface. So these 9 areas. So if you burn, that is the percentage affected by the body surface. So this area contains 18% from the front, 18 from the back. That's about 36% of your body, just your anterior and abdominal area and your trapezoid in the back. This is 1%, your genitalia, 9% from the front and also the back, and 4%. So your arms and hands have about 4% front, 4% in the back. Your head, your scalp, 4% in front, 4% in the back. Your legs, 9% in front, 9% in the back. And your face, 4%, 5% in the back. Skin cancer, we talked about this, is caused by DNA mutation and linked to, we talked about that, the ultraviolet radiation that leads to basal cell carcinoma. And there's malignant melanoma involving the cancer of the melanocytes. We have covered all these concepts, and